In today's lecture, we're going to discuss the process of cellular respiration and fermentation. If you would look at most biochemical pathways, they include multiple reactions of oxidation and reduction. Oxidation is a process in which an atom in some kind of a compound loses an electron, and the reduction is the process in which an atom in a compound gains an electron. Oxidation and reduction are always coupled, meaning they always occur together. Reactions in which processes of oxidation and reduction occur are called redox reactions. When electron is lost, the potential energy of the oxidized compound is reduced. When the electron is gained, the potential energy of the reduced compound is increased. You can see that the transfer of high-energy electrons between various molecules is basically equivalent to the transfer of energy in small increments. Now let's take a look at the process of oxidation using a theoretical and a practical example. In the reaction shown here, A is losing an electron, which means it is oxidized by B. B gains an electron, meaning that it is reduced by A. A functionally reduces B, while B oxidizes A. In this reaction, A serves as a reductant, and B serves as an oxidant. A lowers its free energy because it is losing an electron, while B increases its free energy because it gains an electron. If you would look at the panel on the left, you can see that methane can be oxidized to methanol, and the methanol OH group can be further oxidized to carbonyl group in formaldehyde. Carbonyl group in formaldehyde can be oxidized even further to produce carboxyl group of a formic acid, and formic acid can ultimately be oxidized to carbon dioxide. If you read these processes, these 5 reactions from left to right, you're looking at the processes of oxidation. If you start going from right to left, you're talking about reduction. Electrons within the cell cannot remain free, unbonded, and therefore have to use electron carriers to move from one molecule to another, from the oxidized compound to the reduced compound. Essentially, the function of electron carriers or electron shuttles is to bind and carry high-energy electrons between the molecules, the intermediates, in the biochemical pathways. 3 electron carriers that you must be aware of are NAD, NADH pair, FAD, FADH2 pair, and NADP, NADPH pair. NAD, the oxidized form, shown here, accepts 2 electrons and a proton, becoming NADH, which is a reduced form. FAD, the oxidized form, accepts 2 electrons and 2 protons, becoming FADH2, the reduced form. We're going to talk more about NADP when we're going to cover the photosynthesis. Right now, we're going to focus our attention on NAD, NADH, and FAD, FADH2 pair. So in this situation, the NAD plus serves as an oxidant when it gains an electron, and it serves as the reductant when it loses an electron. Same is true for FAD, FADH2. FAD serves as the oxidant to become FADH2, and FADH2 can serve as the reductant by giving electrons away. You can see where electrons are attached in the molecule of NAD, NADH. So they are attached to this pyridine moiety within the molecule. So, as we mentioned before, the energy currency of the cell is ATP. The cell can't store free energy because that will result in the increased thermal motion of various molecules and may lead to the damage to the cell. So, how do we synthesize ATP? So, ATP is synthesized in the process of phosphorylation. Addition of a phosphate group requires energy and therefore has to be facilitated by enzymes. Within the ATP molecule, phosphate groups are negatively charged. Therefore, they will repel each other. This repulsion makes ATP molecule chemically unstable and very easy to hydrolyze. The release of phosphate groups, namely gamma and beta phosphate groups, I want to remind everyone about that. So this is alpha group, this is beta, and this is gamma. So the release of gamma and, to a less extent, beta groups also releases significant amount of energy. During the ATP hydrolysis, the blue arrow here, ATP is broken down, during ATP hydrolysis, the red arrow here, ATP is broken down to ADP and inorganic phosphate, and energy is released. This energy can be used, as we mentioned before, for the formation of bonds and synthesis of biological molecules, for example, polysaccharides or proteins. Now, ATP is regenerated all the time by reattachment of the gamma, the third phosphate group. So the blue arrow here demonstrates the reaction in which ATP is regenerated. This process is endergonic, therefore, it requires energy. The energy for ATP synthesis comes from the breakdown of monosaccharides, glucose, fructose, or galactose, and we're going to focus specifically on the glucose catabolism. ATP is essentially a link between the exergonic pathways and endergonic pathways in the cell, as you can see in this panel, where exergonic pathways are highlighted by red arrows, while endergonic pathways are highlighted by blue arrows. You can see how ATP can be used in a conversion of a hypothetical compound A to a compound B. The reaction exemplified here is endergonic. ATP provides the energy for the conversion of A to B by the means of some enzyme. As the result, ATP is hydrolyzed to adenosine diphosphate, ADP, and inorganic phosphate, ion. In endergonic reactions, ATP forms an intermediate complex with the substrate and the enzyme at the same time. You can see it here. And it transfers the third phosphate group and the energy associated with it to the substrate, phosphorylates the substrate, and then ADP and inorganic phosphate are released and available for recycling. In the meantime, the substrate is converted into a product due to the energy that was released via the temporary phosphorylation of the substrate by the means of ATP dephosphorylation. One of the pathways for the recovery of ATP molecules is so-called substrate-level phosphorylation. In substrate-level phosphorylation, a substrate carries a phosphate group that is carried to ATP by removing that phosphate from an intermediate via the action of the enzyme. The energy that is released by removing the phosphate from an intermediate is sufficient to add that third phosphate to the available ADP molecule. Substrate-level phosphorylation is not the main way to generate ATP, since most of the ATP is generated by acquiring energy from the catabolism of glucose. Most ATP is produced in the process of chemiosmosis in mitochondria, if we're talking about eukaryotic cells, or on the plasma membrane, if we're talking about prokaryotic cells. Chemiosmosis effectively generates about 90% of the ATP by collecting electrons during the process of glucose catabolism. Chemiosmosis is also used in photosynthesis, as we will learn soon, to generate ATP during light-dependent reactions. Since generation of ATP via chemiosmosis requires oxygen, the process is called oxidative phosphorylation. Oxidative phosphorylation, mediated by the enzyme called ATP synthase, is carried out on the inner membrane of mitochondria. Another component of the respiratory pathway, electron transport chain, is also located on that membrane. As you may remember, mitochondria are double-layered organelles which contain outer and inner membrane, folds called cristae, matrix that is confined within the inner membrane, and the intermembrane space between the inner and the outer membrane. In the next few slides, we're going to cover the process of glucose catabolism, including glycolysis, transition step, Krebs cycle, electron transport chain, and oxidative phosphorylation. The first step of glucose catabolism is glycolysis. It is one of the most ancient biochemical pathways, since all living organisms carry out glycolysis. This universal use of this metabolic pathway suggests that it evolved billions of years ago, and it was the first pathway to break down a monosaccharide, such as glucose. Glycolysis is anaerobic. It always takes place in the cytoplasm, whether it is a prokaryotic or eukaryotic cell. Glycolysis is anaerobic since it does not use oxygen directly. It does not require oxygen for it to happen. The first step, of course, is to transport glucose into the cell. Glucose can enter the cells either via facilitated diffusion using special glucose transporters, or via secondary active transport, usually using sodium glucose cotransporters. The first phase of glycolysis is energy investment phase, shown here. The energy of two ATP molecules is used to convert a 6-carbon molecule of glucose into two 3-carbon intermediates, glyceraldehyde 3-phosphate. You may notice that the number of carbon atoms does not change during the energy investment phase. Then, during the energy payoff phase, molecules of glyceraldehyde 3-phosphate lose energy, not lose, but transfer energy in the form of high energy electrons to NADH, and transfer phosphate groups to 4 ATP molecules, resulting in 2 molecules of pyruvate. You may notice that the pyruvate molecules also contain 3 carbons each. So as we start the pathway with 6 carbons from the molecule of glucose, we finish it with the same number of carbons. Now we distribute it between 2 3-carbon molecules of pyruvate. If we look at the total yield of glycolysis, you can see that 4 ATP molecules are produced at the end, but 2 ATP molecules were used during the energy investment phase. Therefore, the net products of glycolysis are 2 pyruvate molecules, 2 molecules of NADH carrying high energy electrons, and 2 molecules of ATP. ATP molecules are generated at the very last during the last steps of glycolysis by familiar to you substrate level phosphorylation. You can see the illustration right here. One of the glycolytic intermediates, phosphanol pyruvate, transfers the phosphate to ADP, serving as the substrate in the substrate level phosphorylation, resulting in pyruvate and complete ATP molecule. Right now we are at the decision-making point. If the oxygen is available within the cell, then pyruvates that were produced during glycolysis will enter subsequent steps of aerobic respiration. These pyruvate molecules, shown here, enter the mitochondria by the pyruvate carrier protein. This process does require ATP. It is a primary active transport. And eventually, pyruvate ends up in the matrix of mitochondria beyond inner membrane. Within the matrix, pyruvate, this 3-carbon intermediate, is oxidized to acetyl-CoA, which contains 2 carbons originating in the original glucose molecule. And during this process, so this process requires the presence of so-called coenzyme A. You can see the formula of coenzyme A at the bottom. And coenzyme A essentially carries that acetyl group. I'm going to circle it so you understand which group here is an acetyl group. It carries this acetyl group to the Krebs cycle. Acetyl group is attached to the sulfhydryl, to the sulfur atom at the end of acetyl-CoA. During this process, in addition to acetyl-CoA, NAD+ is reduced to NADH, so we get another molecule that contains high energy electrons, and 1 molecule of CO2 is produced, which explains the carbon counting. So this molecule on the left has 3 carbons. The acetyl residue on the right has 2 carbons, because 1 carbon ended up being converted to CO2. Acetyl-CoA enters the Krebs cycle. Krebs cycle is the process that occurs in the mitochondrial matrix of eukaryotes or in the cytoplasm of prokaryotes. As any proper cycle, it is a closed loop. So the very last part of the pathway regenerates what we call the first compound. Of course, you have to take it with a bit of a grain of salt. Which one is the first? So for convenience, we presume that this is the first step and this is the last step. Per molecule of pyruvate, per molecule of acetyl-CoA, Krebs cycle generates 2 carbon dioxide molecule, 1 molecule of ATP, 3 molecules of NADH, and 1 molecule of FADH2, essentially more electron carriers loaded with high energy electrons. This pathway is aerobic. Why? If you look at this pathway, you're not going to see any oxygen here, but NADH and FADH2 ultimately carry these electrons to the electron transport chain, which transfers those electrons to oxygen. Without oxygen, Krebs cycle cannot run. You can see that Krebs cycle produces very small amount of ATP directly and doesn't directly consume oxygen. But again, in order to recover NAD plus from NADH and FAD from FADH2, oxygen at the end of electron transport chain is required. First 3 steps of Krebs cycle form citrate, isocitrate, and alpha-ketoglutarate. If we go back, you can see that acetyl-CoA, which contains 2 reactive carbon atoms in the form of acetyl group, interacts with oxaloacetate, which contains 4 carbons. 2 carbons of acetyl-CoA and 4 carbons of oxaloacetate in the first step of Krebs cycle generate 6-carbon citrate molecule and release coenzyme A. Coenzyme A will eventually combine with another acetyl group and bring it to yet another spin of the Krebs cycle. The reaction of citrate formation is very exergonic and therefore irreversible. It is regulated by ATP levels. When ATP levels rise, the rate of this reaction slows down, and therefore the rate of Krebs cycle and the rate of aerobic respiration slow down as well. During the second step, citrate isomerizes into isocitrate, which is converted during the third step to alpha-ketoglutarate. This is the oxidation reaction. Isocitrate is oxidized, losing 1 carbon in the form of CO2 and forming 5-carbon alpha-ketoglutarate. The electrons that are removed from the molecule of isocitrate are loaded on NAD+, producing another molecule of NADH. This step is negatively regulated by ATP and NADH, meaning that when levels of either ATP or NADH in the cell rise, this process slows down, and therefore the entire aerobic respiration slows down. Conversely, increased level of adenosine diphosphate speed up step 3. In a step 4 of Krebs cycle, 5-carbon alpha-ketoglutarate is oxidized to 4-carbon succinyl-CoA. During that step, the carbon that is lost is released in the form of CO2, and the electrons that were removed from alpha-ketoglutarate during the oxidation of it are loaded on NAD+, producing another molecule of NADH. Succinyl-CoA production is negatively regulated by ATP and NADH, and succinyl-CoA as well. In a step 5, succinyl-CoA is converted into succinate. And during that step, substrate-level phosphorylation forms a molecule of ATP, and coenzyme A is released. In a step 6, succinate is oxidized to fumarate. You can see the formation of a double bond here. And the electrons that are removed from succinate are loaded on FAD together with 2 protons producing FADH2. In the 7th step of the Krebs cycle, fumarate is hydrated, producing malate, and in the 8th step, malate is oxidized to oxaloacetate. OH group is oxidized to carbonyl group, and the electrons and protons that are removed from oxaloacetate are loaded on NAD+, effectively producing another NADH. These 4 carbons then interact with another molecule of acetyl-CoA, starting the cycle again. So one turn of the Krebs cycle processes 1 molecule of acetyl-CoA. 1 molecule of acetyl-CoA is derived from 1 molecule of pyruvate. 1 glucose molecule produces 2 molecules of pyruvate during glycolysis. Therefore, these 2 molecules of pyruvate will be converted into 2 molecules of acetyl-CoA, and therefore will require 2 turns of the cycle per 1 molecule of glucose. First 2 glucose carbons are oxidized and converted to CO2 during the transition step, in which pyruvate is oxidized to acetyl-CoA. Other 4 carbons enter Krebs cycle in the form of 2 acetyl groups, and during 2 turns of the cycle, 4 more CO2 molecules are produced. Essentially, by the end of these 2 spins, or 2 turns of Krebs cycle, all 6 carbons from the original glucose molecule are oxidized and incorporated into carbon dioxide. 1 turn of the cycle produces 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of ATP or GTP. Therefore, per 1 molecule of glucose, 2 turns of the cycle will produce 6 molecules of NADH. This is my typo. I'm going to correct myself here. 6 molecules of NADH, 2 molecules of FADH2, and 2 molecules of ATP. Krebs cycle is said to be amphibolic, which means it can be both catabolic and anabolic, since the intermediates of the Krebs cycle, such as oxaloacetate or alpha-ketoglutarate, can be used to synthesize non-essential amino acids. The final step of aerobic respiration is an electron transport chain, which uses atmospheric oxygen as the final electron acceptor. Electron transport chain consists of the series of reduction-oxidation reactions, at the end of which electrons reduce oxygen molecule, producing water using available protons. The chain consists of 4 protein complexes that transfer electrons between themselves, and these proteins are located either in the inner mitochondrial membrane or in the plasma membrane if we're talking about prokaryotic cells. So what is happening during the electron transport chain? The protein complex 1, shown here, oxidizes NADH and uses energy of these electrons to pump hydrogen ions across the membrane from the mitochondrial matrix into the intermembrane space, increasing the hydrogen concentration within the intermembrane space and decreasing it within the matrix. Complex 2 oxidizes a molecule of FADH2 to FAD. The electrons that are removed from FADH2 are transferred by the means of ubiquinone B, Q, to the complex 3, which uses the energy of these electrons to again pump hydrogens from the mitochondrial matrix into the intermembrane space across the inner membrane of mitochondria. And finally, electrons, by the means of cytochrome C and the 4th complex, are transferred to oxygen, which, when combined with 2 hydrogens, form a molecule of water. The complex 4 also uses the energy of the electrons to pump hydrogens across the inner membrane of mitochondria. All this pumping of hydrogens effectively results in the formation of the ion gradient, the gradient of hydrogen ions. We call this gradient an electrochemical gradient. So electrochemical gradient essentially is formed by the use of the energy of all the redox reactions that we were talking about. Energy of the electrons that were collected during glycolysis, energy of the electrons that were collected during transition step, during Krebs cycle. All those electrons, energy of those electrons is used during the in the electron transport chain to pump hydrogen ions, protons, across the mitochondrial membrane. In other words, proteins of electron transport chain oxidize both NADH and FADH2, or are reduced by both NADH and FADH2. This uneven distribution of hydrogen ions, as I mentioned before, is known as an electrochemical gradient. This electrochemical gradient, the gradient of both the concentration of hydrogen ions and the charge, the accumulation of positively charged hydrogen ions, makes the intermembrane space more positive, while matrix becomes more negative. This electrochemical gradient drives the diffusion of hydrogen ions by the protein called ATP synthase. As you can see, hydrogen ions are positively charged. And as you may remember from the material in the week 5, positively charged ions cannot cross the membrane directly and have to pass through a channel. ATP synthase essentially serves as the channel for hydrogen ions to move. So the potential energy of this gradient drives the movement, the diffusion of hydrogen ions through ATP synthase. And the kinetic energy of that diffusion is transformed by ATP synthase into the potential energy of the chemical bonds in the molecule of ATP. The energy of hydrogen ion diffusion essentially provides the required energy for the ATP synthesis from ADP and inorganic phosphate. This table represents the summary of aerobic respiration. During glycolysis, 2 NADH molecules are produced along with 2 ATP. During transition reaction, another 2 NADH molecules are produced. In Krebs cycle, 6 NADH and 2 FADH2 molecules are generated along with 2 ATP. So the total yield of all these reactions is 10 NADH, 2 FADH2, and 4 molecules of ATP. This is what's produced when 6 carbons of glucose are converted to 6 molecules of carbon dioxide. Now, 1 molecule of NADH carries enough energy in their electrons to produce 3 molecules of ATP. Therefore, 2 molecules of NADH will generate 6 ATP molecules. 1 molecule of FADH2 carries enough energy in the electrons, in the form of electrons, to generate 2 ATP molecules. Therefore, 2 FADH2s will be enough to generate 4 ATP. Via this fairly simple calculations, you can see that the total number of ATP produced during oxidative phosphorylation and electron transport chain is 34. Combined with 4 ATPs produced in Krebs cycle and in glycolysis, the maximum theoretical yield is 38 ATP molecules. Now we need to answer a couple of important questions. First, why is the practical yield is lower than the theoretical yield? There are 2 reasons. One, transport of intermediates, such as pyruvate, from cytoplasm to mitochondria requires energy. And second, when we say that hydrogen ions cannot cross the membrane directly, it's not entirely true. Membrane is a little leaky for hydrogen ions. So some of the hydrogens that end up in the intermembrane space manage to bypass ATP synthase and essentially cross the membrane directly, which explains why energy of some of those hydrogen ions, the potential energy of the gradient, is not used in its entirety. Why does FADH2 provide electrons for synthesis of only 2 molecules of ATP, while NADH for the synthesis of 3 molecules? If you would use the points in electron transport chain where NADH and FADH2 are oxidized, you may notice that the electrons from NADH are removed and enter first complex, then ubiquinone, then third, then fourth, and then finally are added to oxygen. The electrons from FADH2 enter second complex, so they start a little bit later. If you will think about the electron transport chain as a ladder, and electrons as a ball, electrons from NADH start at the top of the ladder, while electrons from FADH2 start 1 step lower, therefore generating less energy. Now we're going to move on to talk about other concepts associated with glucose catabolism, namely anaerobic respiration and fermentation. So, electrons at the end of electron transport chain are very dangerous to the cell. Electrons can literally damage a lot of chemicals. So electrons are received by the final electron acceptor. In aerobic respiration, and as we figured out, the final electron acceptor is oxygen. In anaerobic respiration, the final electron acceptor is inorganic molecules that are not oxygen, for instance, sulfate or nitrate. You can see nitrate as being final electron acceptor in the process of anaerobic respiration carried out by bacteria called Pseudomonas tutseri. It's important to highlight that both aerobic and anaerobic respiration include the steps of glycolysis, Krebs cycle, and electron transport chain. However, electron transport chains for aerobic and anaerobic respiration have different enzymes. Functionally, they do the same thing. They transfer electrons from electron carriers to the final electron acceptor, but the enzymes that are involved in the process are different. There are some organisms that can actually carry out both aerobic and anaerobic respiration. Fermentation, anaerobic process, uses organic molecules as the final electron acceptor. For example, acetaldehyde in the process of alcoholic fermentation, or pyruvate in the process of lactic fermentation. You may ask, why do we even need a final electron acceptor? Well, if you think about the role of NAD plus NADH and FAD-FADH2 pairs, their role is to simply carry the electrons. So they don't have to cling to them forever. All they need to do is to carry the electrons from each step of glucose catabolism to the electron transport chain, where they have to relieve them. If there is no electron transport chain involved, they still have to give those electrons to final electron acceptor to be able to collect electrons again. They essentially work as some kind of an electron taxi. So fermentation occurs in the absence of oxygen, and it's an incomplete oxidation of carbohydrates. As we mentioned before, the final electron acceptors for fermentation are organic compounds. Fermentation produces much smaller amount of ATP than respiration, and speaking of respiration, if we compare two, aerobic and anaerobic, aerobic respiration always produces more ATP than anaerobic. So there are bacteria that can use aerobic respiration when oxygen is available, and fermentation when oxygen is not available, and actually they can grow equally fast using either of these processes. Two fermentation processes that you must know are lactic and alcoholic fermentation. During lactic fermentation, at the end of glycolysis, you get 2 NADH and 2 pyruvates. These pyruvates serve as the final electron acceptor to produce 2 molecules of lactate or lactic acid. And NADH is a reductant in this reaction. It is oxidized by pyruvate to 2 molecules of NAD+ that can be used during glycolysis to collect more electrons. So the ATP yield of lactic fermentation is 2 ATP, just like in glycolysis. During alcoholic fermentation, pyruvate is first oxidized to acetaldehyde, producing carbon dioxide. And then acetaldehyde is further converted by the enzyme called alcohol dehydrogenase to ethanol. NADH serves as an electron donor to reduce acetaldehyde to ethanol, producing NAD+, which again can be used in the process of glycolysis as the oxidized form of an electron carrier. We all know that processes of alcoholic and lactic fermentation are widely used in food industry. Lactic fermentation is used to produce a bunch of different dairy products such as buttermilk or yogurt, as well as sauerkraut and other pickles, and you can name multiple foods that use lactic acid fermentation. What if carbohydrates are not available? Well, in this case, triglycerides or proteins can be used as the energy source. The pathways that break down proteins, carbohydrates, and triglycerides are all interconnected and often are reversible. For instance, intermediates of carbohydrate catabolism, intermediates of the Krebs cycle, can be used in the synthesis of other biological macromolecules such as steroid hormones or amino acids. Now, what's going to happen to triglycerides during their catabolism. First, they're broken down to glycerol and free fatty acids. Glycerol is converted to glyceraldehyde 3-phosphate, which is the intermediate of glycolysis and can be converted to pyruvate. Pyruvate can then be converted to acetyl-CoA, which enters the Krebs cycle, and the rest is history, you know that. Fatty acids can be converted to acetyl-CoA in the process of beta oxidation, and as we mentioned before, acetyl-CoA can enter the Krebs cycle. Beta oxidation, just like a Krebs cycle, occurs in the mitochondrial matrix. Proteins will be catabolized to amino acids, and amino acids will enter two distinct processes, either transamination or deamination, resulting in keto acids. These keto acids, such as pyruvate, can be converted to acetyl-CoA, or some of them can enter a Krebs cycle and supply and generate more energy through this catabolic pathway. In the next conversation, we're going to touch upon the process of photosynthesis, which has certain similarities with cellular respiration.